Okay, so for this class tonight, we're going to finish 10, do the bulk of 11, and then Monday is exam 5 over 9, 10, and as much as we cover in chapter 11 tonight, there won't be anything on chapter 11 that we haven't covered. Okay, so it's not going to cover things that are above and beyond what's in chapter 11. Wednesday then will be your review day. So Wednesday, if you have any questions about anything on that review that I just put out for you to be able to pick up, we can run through problems, questions, anything that you want in terms of practicing, making sure that you understand the kind of question that you're going to expect for that final. And then that puts that following Monday for the final. All right. So the last thing, we have basically gone through all of this. We talked about the 7 of the 8 functions that proteins have. So remember, like the first 3, how you have insulin. So those are the messengers. They bind to receptors. Those are on the surface of the cell. Those are proteins. And then that will affect channels, which are proteins embedded in the membrane that will allow glucose to come in. So like this one slide has 3 functions of proteins on it. Then you can also have the carriers. So carriers have the job of transporting things from one place to another or holding on to those substances. Hemoglobin's a good example. Hemoglobin is the protein in your red blood cells that binds oxygen. 5 was protection. Good example of protection, antibodies. So antibodies are made by white blood cells. When your body is infected by a virus or a bacteria, white blood cells figure out that there's a foreign substance that's present and they begin to make a protein. The protein, antibodies, stick to the foreign cell or the infected cell, and that marks that cell as foreign. That attracts more white blood cells, and it attracts specific white blood cells called macrophages. Macrophages are the ones that look like they go and they like eat stuff. So they'll like swirl around and actually engulf foreign substances and destroy them. So antibodies don't actually destroy foreign things, but they actually help to mount and stimulate an immune response. Then 6 and 7, so these 2, we talked about contraction. So this is how there are proteins inside of all of your muscle cells, whether you're talking about skeletal, smooth, or cardiac muscle. That's the 2 muscle proteins are actin and myosin. Actin is the thin-looking string. Myosin is the thick-looking one. And they form like an overlap of layers. So you'll have a layer of actin and a layer of myosin, and they overlap slightly. So it's sort of that little drawing on the left. But what they do when they're stimulated is they pull on one another. So they literally pull on each other and overlap more and more, which causes the muscle cell to shorten. 80% of the interior of your muscle cells are made of actin and myosin. And if you build up, if you like work out and you lift weights, you'll notice that your muscles get thicker. They get thicker, your muscles get bulkier because there's more actin and myosin proteins that are built and added to those muscle cells to make them stronger. 7 is the anchoring. So these are those fibrous proteins that we talked about. So collagen, the most abundant protein in the body. Collagen, remember, it looks almost like string. It looks like thread, like what you would sew with. So it's actually multiple strands of proteins that are wrapped in a very stringy, strand-like arrangement. I always think of collagen, I think of like tendons and ligaments. Tendons help to anchor muscles to bone. Ligaments hold bones to bone. So they help to anchor all of the skeletal structures and the muscular structures onto the skeleton. All of your connective tissues, so if you've taken A&P, when you think of connective tissues, you think of bone, cartilage, dense, regular, irregular connective tissues. All of those anchoring kinds of tissues all have collagen. Some of those anchoring tissues also have elastin, and elastin is stretchy. So elastin is the reason that when you lift and pull on your skin, it drops right back down because of elastin. And so it gives you this sort of stretchiness so that skin and other tissues can get stretched, but they actually bounce back. But unfortunately, when you get old, they don't bounce back like they used to, so they stretch and they stay stretched. And that's why the skin then starts to wrinkle. Why you end up having folds in the skin is because of loss of elastin over time. But then that leads us to 8. So the last one is an enzyme. We've talked about enzymes. We talked about enzymes in Chapter 5 mentioned them again. So remember, in talking about how you speed up rates of reactions. So remember that typically enzymes are proteins. So enzymes have a specific shape, so they can fit and interact with the reactants in a reaction. They act as these catalysts because remember, they lower the activation energy. So they do this by being able to, and this image is a pretty good one. So this one, you can kind of see here, the shape of this enzyme. The green is the enzyme, like a three-dimensional. Remember, they look globular. But then the reactant in this, or substrate that this enzyme interacts with, is glucose. So glucose is the little black and red balled structure that's kind of down in the little crevice. So I think of enzymes kind of have this shape, and the substrate or the reactant fits into that little crevice because of the shape. And that allows this reaction to speed up. So the reaction up here, where glucose combines with ATP, it would happen anyways, but the enzyme hexokinase speeds it up so that reaction is going to take place really fast by lowering the activation energy. Okay, so things to remember. Remember that, nope. Okay, so remember that the reactant is the substrate, means the same thing. Where the reactant sits is called the active site. I just think of it as the cleft that the reactants come together in. And just remember that it helps to lowers the activation energy. So the reaction happens super fast. Enzymes can speed up reactions 1,000,000 times faster than if you didn't have the enzyme alone. So this was really critical for us to become super complex people. Living organisms that have more than one cell, we have to have enzymes so that we can respond to changes in our environment and maintain homeostasis. Good example, you go outside and it's cold, and what do you automatically start to do? Shiver, okay? You get goosebumps. So the response, the only reason that the response can happen this quick is because of enzymes. Enzymes that trigger changes to help speed up reactions so that you can kind of counter what's going on outside of your environment so that you can try to maintain balance. So this is completely like review. Remember in chapter 5, we said that enzymes bring reactants together in the right orientation, and that's going to help to speed up that reaction. It helps to make that bond easier to break or easier to form. And so notice there's the activation energy without the enzyme is this high. But when I add an enzyme, now my activation energy is a lot lower because of what the enzyme is able to do. Now we can still change how fast enzymes work by changing a couple of the criteria. So one, and I actually put a couple of slides together on this one. So if I have an enzyme and I want it to work faster, I can add more reactant, right? So if A plus B makes C, if I take a lot of A and B, then the enzyme is going to work as fast as possible because it has enough building materials, right? But if you run out of A or B, then you can't make C no matter what. But substrate concentration, the amount of materials is critical. And I kind of liked in the book, they give the little analogy of the bicycle makers. So it's saying if you have these three guys and they're all in help, they're the enzymes, they're the workers. And so as long as you give them enough parts, they can make a certain number of bicycles a day. So it's a matter of like just how fast they can work. They can only work but so fast to make them. You could give them enough bicycles to make, or enough parts to make 100 bicycles, but they might only be able to make max 25, right? But if you don't give them enough to make 25, then they're not going to be able to do anything. They're going to sit around because they don't have the parts. So that's the substrate concentration. The second one, though, is one that we didn't really talk about in chapter 5 because we hadn't really talked about acids and bases. So this is one that now kind of brings in this idea about chapter 9, and that's pH. If you change the pH, that is going to change the amount of hydrogen ions around the enzyme. And that can change the enzyme's shape. So if you change the pH, then remember that is going to change the amount of hydrogen ions, that H3O plus or just H plus. And if you change the ions around the enzyme, the result to that is that can change the shape of the enzyme. So maybe you remember that when we did the antacid lab. So we actually tried to digest lactose under an acid concentration, a base concentration, and then neutral. And there was a very defined result, like the acid, nothing happened. Everybody was like 0, no glucose, didn't work, okay? So under an acidic condition, that enzyme was not folded correctly, and if it doesn't fold right, then it's not gonna work. The other one that we did kind of mention was temperature. And this is one, remember, we tested digestion under ice water, room temperature, body temperature, and even boiling water. So we did all of them and found that different temperatures ended up having different enzyme activities. So typically, there is an optimum. Most things that happen in the body, the temperature that's optimum is 37 degrees Celsius, right? Because that's body temperature. One example of this, though, sperm production. So sperm production actually does not happen at an optimal temperature of 37 degrees or 98.6. It happens best around 35.5 degrees, which is about 3 degrees Fahrenheit below normal body temperature. So optimal sperm production is usually about 95. So where are the testes located? Are they inside of the body? They're outside of the body, and that actually lowers their temperature. So that is the reason for it. So, and in fact, the external temperature can affect where the testes are located. So I don't know if you've ever seen bull cows in the summertime. Okay, so bull cows, so their testicles are really pretty big. But when it's hot out, they will hang way down. I mean, they'll hang down like this. The reason is because it keeps them cooler. When it's cold, they get pulled up close to the body, and that keeps them warmer. And it's all because sperm production takes place 3 degrees below normal body temperature. That's why they're kept outside of the body. So it's just an example. Most enzymes, optimal is right at body temperature, but that's kind of an odd process, and the reason why they're external. So this one actually shows some of the differences in pH. pH does vary in terms of enzymes in the body. Some pHs, like urease, which is in the liver, pH is pretty close to neutral. But then pepsin, which is in the stomach, pH is really acidic, but that's because the environment of the stomach is acidic. And so this enzyme has kind of adapted so that it works better under an acidic condition. So most temperatures are about the same for body enzymes, but the pH can vary depending on where the enzyme actually is. So then the last thing. So we have the amount of starting materials, the pH, the temperature. The last thing is what they call inhibitors. And you can kind of think of them as being like the fourth thing that can affect an enzyme. So when you think of an inhibitor, what does an inhibitor do? If something inhibits you, it what? Blocks you, right? So when something inhibits, it's sort of a blocker for you. And that's exactly what these are. Inhibitors block an enzyme. It's going to prevent the enzyme from actually speeding up the reaction like it should. Some inhibitors are reversible, and some of them are permanent. So irreversible, an irreversible inhibitor, it will bind to the enzyme permanently. And block it so then it's basically done. But if it's reversible, Reversible inhibitors mean that the inhibitor can bind to the enzyme, but then it can come off. So it's almost an equilibrium reaction. So it's not permanently covalently attached to the enzyme. And a reversible inhibitor, if it comes off, the enzyme can start working again. So it just depends on if it's present and if it's attached. So these, the ones that are reversible, the ones where you can have them attached or fall off, they can actually be what is known as being competitive or non-competitive. So if you're competitive, what do you think of that? What do you think when that means? Like if something competes, if it's competitive, what's that word mean to you? Okay, so going against something like directly competing, so with something else. And so that's exactly it. So this, if the inhibitor is competitive, it tries to bind the active site. So it wants to bind to where the substrate should bind. So it's going for the same spot on the enzyme. And so if the inhibitor gets into the active site, then the substrate can't. So the reaction's blocked. So like I always think of the enzyme like forming this little cleft, and that's where the substrate or reactant fits. If the enzyme sits there, as long as the enzyme is there, the reaction's going to be blocked. But if it's non-competitive, this means that the inhibitor binds somewhere else on the enzyme. But what it does is it changes the shape of that enzyme. So this binds to the enzyme and it changes the shape of the active site. So I have all three. I actually took all three of these and put them all on one page so that you can actually kind of see, compare and contrast them together. Okay, so this is the first one. So in this first one, E is the enzyme, S is the substrate. So by substrate, remember that all I mean is it's like the reactant. So it's the thing that the enzyme wants to react with. So this is the substrate or reactant. And then here, this is my inhibitor. So the little green thing is the inhibitor. Okay, so in this one, here's the normal reaction. This is the normal reaction. So normally the enzyme's sitting there, the substrate binds, the reaction takes place, the product is released. So here's your product. So this is like the normal reaction if there's no inhibitor present. So what does this one do? Would you think this one is, so notice if the inhibitor comes into play, see how it blocks the substrate? Is this a competitive or non-competitive? Is the inhibitor binding where the substrate binds, or is the inhibitor binding somewhere else? This one would be considered non-competitive. Because what happens when the inhibitor binds? Do you see the shape change? So notice how the active site, so remember this right here, they call this the active site. That's where the substrate sits. So normally the active site, in this example, the active site looks like a little sharp point. But when the inhibitor binds, look at what happens to the shape of the active site. Now it rounds out. And if it rounds out, the substrate doesn't fit anymore. So it's almost like changing the lock. Right? It changes the shape of the lock and the key doesn't fit anymore. So that's a non-competitive inhibitor. So this is what happens with lead poisoning. So remember we were talking about lead poisoning and how it tastes, that lead chips taste sweet, and that's why like little kids eat lead paint chips. So lead is a non-competitive inhibitor. So what it does is it binds to the enzymes that are involved in producing normal nerves, like nerve development, neuron function, neuron structure. So there's enzymes that are all taking place during infancy, childhood, all the way up into your early 20s. You have lots of this neural development. And lead actually binds to the enzyme, changes the active site. So now the enzyme can't provide or promote that development. So lead poisoning does this. But that does mean what you can do is, as long as you remove the lead from the environment, then over time you'll have less and less lead in the body, because notice it is an equilibrium reaction. So sometimes the inhibitor comes off. So in this, sometimes the inhibitor is on, but the inhibitor is not permanently attached. So making the remedy of removing the lead from your environment, over time, the amount of lead will be decreased and you won't have the negative side effects. So that's a non-competitive inhibitor. Compare this with the one on the far right. So here's the normal, right? So normal enzyme binds the substrate, makes the product, right? So that's the normal. From top to bottom, the reaction goes down. But then look, if you go the enzyme, if the inhibitor binds, how is that different than the first one? The inhibitor has the same shape as the substrate. So when the inhibitor binds, the substrate can't. So this is an example of what? Yep, this one is competitive. So it competes. It competes with the reactant or the substrate. It sits down and that's the block. As long as it's there, the substrate can't bind to it. But again, notice that this is an equilibrium reaction. So sometimes the inhibitor comes off. And if the inhibitor comes off, then the substrate can bind. An example of this is what happens with carbon monoxide poisoning. So carbon monoxide doesn't necessarily bind an enzyme, but it binds hemoglobin. And when it binds hemoglobin where oxygen normally sits. So remember we were talking about hemoglobin and we were talking about the shape of it. And I said hemoglobin carries oxygen because of those iron atoms. Carbon monoxide comes in and sits right where the oxygen should be. And so no oxygen can get picked up. So that hemoglobin's basically blocked. And so it actually binds hemoglobin better than oxygen does. And so this can lead to depletion of oxygen in the blood. And that's why they suffocate and die, like lose consciousness, all the toxic effects that carbon monoxide can have. But it's a competitive inhibitor. Carbon monoxide kind of like wants the spot where oxygen should be. Whereas lead binds somewhere else on the enzyme and changes the shape of the enzyme so it can't work. But both of those, notice the arrows go back and forth. Both of those are reversible. The middle one, though, look at the arrow in the middle one. So this one, the arrow goes one direction. So that means this is the irreversible inhibitor. This is the one that you can't once it binds, you're done. The enzyme's done. The enzyme can't do anything else. Inhib. So see how it looks like the inhibitor like binds to the active site permanently? And this is a permanent thing. So that enzyme can't function anymore at all. See the arrow? So here, do you see the arrow goes up, forward and back? See over here, the arrow goes forward and back. So that means the inhibitor can come off or go on, come off and go on. But if you look at this one, notice that the arrow goes only in one direction. That's why it's irreversible. That means that once the reaction happens, the inhibitor binds and the substrate can't ever bind again. So this one's a permanent. Basically, this enzyme's done. So interestingly, this goes into this last slide. Antibiotics, in many cases, are actually inhibitors of enzymes that bacteria and viruses use. And so one that was found was penicillin. It's kind of interesting. Penicillin was found by accident. It wasn't like they didn't research it and discover it. It was actually found by accident because this was right when they were starting to grow bacteria. So there was a scientist and they grow bacteria. It's on this like jello kind of surface. So it's got broth, it's got salts, it's got some nutrients in it, but it's kind of got gelatin in it. It's called agar, but it feels a lot like jello. And so it's this surface. What they found is if they spread bacteria on this surface, the bacteria could pull the salts, water, nutrients out of the agar and grow. And you can end up then, like now you have a little culture of bacteria, so you can study it. So early scientists, this is really how they maintained bacteria cultures. So they would grow like Staphylococcus, Streptococcus, E. coli, so they could grow them on different plates. So this scientist had his plates and he went off for the weekend and came back thinking that his little plates were going to look perfect. And when he came back, he found that there was mold, that there was a spot of mold growing in the middle of the plate. And so he was like, oh, this is contaminated, because mold and bacteria are two different things. And so he went to throw it away, but he stopped because the bacteria, the part that I kind of shaded in purple, that part here, this is the bacteria. And it grows on the surface of the agar and makes like this solid layer. But he found where the fungus was, where the mold was growing, there was no bacteria. So he stopped and looked at it and said, this fungus, this mold, has to be making something that inhibits the bacteria from being able to grow, because they can't grow up to it. There's like a ring. There's like a zone where nothing grows. And so he was like, got really interested and said, okay, let's study this. Why? What is this mold doing? So then he started growing mold and actually trying to test it with different bacteria and found the same thing. He came up with the term antibiotic, antibacteria, because it stops the growth of bacteria. So then they isolated it, and this chemical is penicillin. So this was one of the first antibiotics that was used. It was originally grown from molds, a very specific mold. They named it penicillium, the mold, because it makes penicillin. And what penicillin does, it's an irreversible inhibitor. So just a little bit about bacteria cells. So bacteria cells are single-celled. They don't form large groups. They don't have to live with other bacteria. It's just a single individual cell. So if I was a little bacteria cell, this is what I might look like. They're unicellular. So because of that, they can grow on your hands. They can grow like on the door handles. They can grow on the desk. They're everywhere. They grow on like little dust bunnies under the bed. They're pretty hardy little things. One of the ways they're really hardy is they actually make this really thick cell wall. And that's almost like this extra cover for them, and it protects them. This keeps them from drying out. It keeps them from being affected by changes in salt and nutrients and everything else. They got it's almost like their own little wall, this walled cover. Penicillin blocks formation of the cell wall. So if you think of this cell wall as looking like a chain link fence, So that's kind of like this little 8 blocky looking thing that I have on the left. So in order for a cell to grow, it has to make the cell wall. So the cell starts off this big, it actually grows and gets longer, and then it'll actually pinch into 2. And then those 2 cells will grow, and then they'll pinch into 2. So they keep on growing and dividing. As long as there's nutrients present, bacteria grow happy. But they have to make cell wall then, because if for the cell to go from being little to being big, it's going to have to increase the amount of cell wall it has. So what it does is it takes the structures of the cell wall and assembles it into this kind of block-like layering for protection. Penicillin blocks the formation of the links. So penicillin will turn around and block this, and block this, and block this. And so it stops these links from forming, and what you end up with is that. So does that look like a wall? It has a lot of holes in it, right? So it ends up having, it's really weak and has a lot of holes, and the cell keeps growing, and it literally grows out of its cell wall. The cell wall just like breaks apart, falls off. But once the bacteria loses its cell wall, now it's subject to all kinds of damage, because now anything else around, salts can cause changes to it. Any other microbes out there can attack it. Fungus can completely destroy it. And so by blocking the formation of the cell wall, that has an antibacterial effect. So it was really kind of amazed, like, oh, this is how it works. So then they started growing fungus. So they started growing mold, and then extracting penicillin. And then they started, initially penicillin was only given by injection. So you would grow the mold, they would extract the penicillin, they had to sanitize it, but it was always given as an injection before they finally made up the oral pill form. But they had to go through and study. 1, they had to understand bacteria. 2, they had to understand what the fungus was, and what the fungus was doing is actually blocking the formation of a bacteria cell wall. So this was, again, something early turn of the century, early 1900s. Okay, so 1910, 1920 is when when penicillin really kind of came on the market. Penicillin is other antibiotics actually attack the interior of the bacteria. So there's other antibiotics that block enzymes that only bacteria use. So then that's one of the reasons why I like talking about the whole virus thing, because development of a lot of medications, what you do is you have to understand all of the life cycle and the enzymes that are involved in the life cycle of a virus or of a bacteria or even of a fungus. And then you look for what enzymes do they do that we don't use, because if we can block that enzyme, then it's going to block their ability to grow. Right? So taking penicillin, you don't make cell walls. So if you take penicillin, penicillin's not going to block your cell walls. Unfortunately, what does penicillin sometimes do in antibiotics? It doesn't affect your human cells, but it affects all the little microbes that live in your body. Right? So a lot of times people that take penicillin or take an antibiotic, you get a yeast infection. And the reason is because the antibiotic actually damages the bacteria that live in the female reproductive tract. And so you damage those bacteria, and that allows the yeast to grow like mad. And that's why they say you have a yeast infection. So normally they're in balance. Normally you have bacteria and yeast. They live together. Okay, but if you knock out all the bacteria, then the yeast grow, and that causes all the negative side effects where you're oh, miserable person. Okay, but it's because penicillin doesn't cause any damage to your cells, but it can cause changes to bacteria cells. I don't know if you've ever taken an antibiotic and it causes diarrhea. Tears your GI tract up, and so then you're I can't take this anymore. This is and that's because you have all those bacteria that live in your GI tract. So the antibiotic can affect their function as well, because most antibiotics are just triggered, they're just targeted towards bacteria. Okay, but this is a good example of this. Okay, so penicillin. Penicillin is an irreversible inhibitor. So it binds to the enzyme and the enzyme's done. The enzyme can't function anymore because it doesn't come back off. It's an irreversible inhibitor. Carbon monoxide is a competitive inhibitor. Lead poisoning is a non-competitive inhibitor. But both of those, they can come off of the enzyme or beyond. So they sort of like go back and forth. All right. So that is the end of 10. So 11. 11 is nucleic acids. So nucleic acids, we're talking about DNA and RNA. So one thing that I like to do at the beginning of this chapter is kind of think about DNA versus RNA in comparison to all of the other nutrients and molecules we've been talking about. So if we went on a size comparison, the smallest of all of the molecules, the nutrient and biology molecules, the biomolecules that we're talking about, the smallest of them are the simple sugars, right? So in this, remember, we're talking about stuff like glucose, sucrose, table sugar, fructose, fruit sugar, maltose, lactose. Those little sugars, the mono and disaccharides, they are the smallest compared to all the other nutrients. If we go up one step, it's a pretty big step because the next ones are lipids. In lipids, whereas simple sugars have anywhere from 25 to 50 atoms, lipids are now in the hundreds of atoms. So remember, so lipids, so your fats and oils, they're bigger than sugars, but they're smaller than all the others, those phospholipids. Much bigger than the lipids are your proteins. So remember that you can talk about things like hemoglobin, collagen. So these are actually now thousands of atoms in size. So instead of under 100, then we have 100 or so. Now this is like 1,000 atoms in size. So we're talking about getting even bigger. So of the remaining, we have the nucleic acids are bigger than any of those. And the only one that rivals that are those polysaccharides. So RNA is typically a couple of million. So these are millions of atoms. Whereas hemoglobin is maybe thousands. Lipids are maybe hundreds of atoms. And the simple sugars are less than 100 atoms total. So RNA is a structure that's made specifically so that you can make proteins inside of the cell. RNA is not always present. It's only made as it's needed. So it's actually smaller than most of the complex carbohydrates. Complex carbohydrates, that's like starch. Like starch, like fiber, cellulose, glycogen if you're talking about human. So this, now we're talking about billions of atoms in one molecule. But that still doesn't hold a candle to DNA. DNA is in the trillions, like hundreds of trillions. So it's huge in comparison to all the other molecules when you think about like water and salts, then sugars, then you build up to the lipids and proteins. So then you have the complex carbohydrates, but DNA is like bigger than any of them all combined. Okay, so this, we're talking about our genetic information. And it is literally trillions of atoms in size for one molecule of DNA. So the thing to remember is that nucleic acids, being considered this genetic material, Nucleic acids contain all the information that is needed for you to go from one single fertilized cell, right? So you had an egg from your mama, sperm from your dad. They joined, united, making this unique set of genetic information. You're like a hybrid. You're a hybrid of the mother and the father genetic information. That comes together to make this new combination that's you. But in that genetic information is all the information the cell needs to know in order to divide, in order to form layers, in order to form tissues, in order to form nerve tissue like brain, in order to form skin, the surface and covering, all that development. So it controls everything from growth, development, from one single cell all the way to you, and you're actually made up of a couple trillion cells, which is 1,000 billion, right? So it goes million, billion, trillion. Along the way, proteins to do all this stuff that we just talked about in chapter 10, to make proteins. All that information and the instructions are all contained within the nucleic acids, within the DNA. So protein production, like if you need to make hemoglobin, if you need to make actin or myosin, if you need to make the 20,000 different enzymes that your body makes, all that information is contained within the DNA. So protein production, cell division, what each cell does, because like your skin cells, they make keratin and form this little waterproof layer on your skin surface. But then red blood cells develop and all they do is carry oxygen. Muscle cells, their big job is to contract. Nerve cells send nerve impulses. So the sort of specialization of these cells, all that is all contained within DNA. It's pretty it's like the blue book of everything, like Wikipedia. it's the Wikipedia. It used to be the encyclopedia, but nobody knows what those are anymore. So it is like the Wikipedia where every single possible topic related to you is all contained. So when we talk about DNA, it's always found in the nucleus of the cell, right? So if you look at a generic cell, there's always this little round circle in the center, like looks like the yolk of the egg, right? There's always this little circle right in the center. That's always the nucleus. In fact, there's DNA in other places, but that's really the primary one. The sequence of the DNA is what forms what they call genes. Okay, so the actual order of the sequence, just like remember proteins, the amino acid order was that primary structure. So we're going to see that's the same thing. The order of the nucleic acids determines all these characteristics and all the things that your cells end up doing. It is estimated that in your DNA, there's about 20,000 different genes. Some cells use certain genes, some cells don't. Hemoglobin or red blood cells use the hemoglobin gene a lot. Your skin cells, they use the keratin genes a lot to make that waterproofing cover. So let's look at the shapes, like what makes them up. All nucleic acids are made of a building block that is called a nucleotide. So just like proteins are strings of amino acids, nucleic acids are strings of nucleotides. 3 things in a nucleotide. Nucleotides have a nitrogenous base, a sugar, and a phosphate. So a sugar, a phosphate, and then a nitrogenous base. So here's sort of like the whole piece. There's a nucleotide. So the question always comes up, okay, I know what a sugar is. Okay, I know what a phosphate is, right? Phosphate, remember, is that PO4, the phosphate polyatomic ion. The sugar varies. DNA has deoxyribose, RNA has ribose. But this nitrogenous base thing, so nitrogenous bases, they're described and called a nitrogenous base because they're ring structures that have nitrogen. So that's how that name came about. They can act as proton acceptors, which is why they call them bases, but they're really just these ring structures with nitrogen. There's 5 possible types. 3 of them are found in DNA and RNA. These 3, adenine, guanine, cytosine, all found in DNA and RNA. So those are kind of similarities. Now adenine, they almost always just write it as an A, but there's its name. So they'll just put an A. Guanine, there's its name for spelling purposes, so you can recognize it. So they'll usually just refer to it as the G. And you notice they're really similar in shape, similar in structure. They're those double rings. Then cytosine. So cytosine actually looks more like the other 2. The other 2 are unique. DNA only has thymine, RNA has uracil. So all of them, both of them have A, C, and G, but DNA has T or thymine, RNA has U or uracil. So we'll come back and kind of make this compare contrast on a slide like 4 slides from now. The other difference, sugars. DNA and RNA do not have the same sugar. Remember these look like, these are those aldo pentoses. They're only a ring of 5, so if you count, you only see 5 carbons in the ring. They were an aldehyde, so an aldopentose. The only difference is one has an oxygen, one doesn't. So ribose is the kind of normal sugar. Deoxyribose, notice it's missing an oxygen on carbon number 2. That's it. Other than that, they're exactly the same. But this is a difference between them. In fact, this is the R in RNA and the D in DNA. So RNA is ribonucleic acid, so that's the reason it's called ribo. DNA is deoxyribo, which is the reason why it's the D. So if you can remember its name, you can remember the sugar. So these ones get put together. So I said you have a phosphate, a sugar, and a base. So can you see, here's the phosphate, here's the sugar, here's the base. So the nucleotides always have a phosphate, a sugar, and a base. But they get linked together in order to form the DNA or RNA. They get linked into a chain, just like proteins, where the amino acids get linked together. These nucleotides get linked together as well. So here's how they come together. So this one, so this shows there's two nucleotides. So there's a phosphate, sugar, and a base, phosphate, sugar, and a base. So what ends up happening is condensation. So just like how you make the amino acid, make the peptide bond, here there is a link from the hydrogen off of that sugar, hanging off of the 3rd carbon. Remember, it's a little 3. So hanging off of that 3rd carbon, a hydrogen gets pulled off. Off of the phosphate, an oxygen gets pulled off. So there's an H and an O. The other H actually comes from the solution. Okay, so you add a hydrogen as well. So H and O make, what does H and O makes? Water. So notice over here, see water becomes a product. So there's the blue, H and O, and they get, now there's a link. So here's the link between the 2 nucleotides. It's a condensation reaction, just like what you see when amino acids link together. All this is doing is it ends up linking the sugar to the phosphate on the next nucleotide, and then this sugar gets linked to the next one. So you end up having sugar, phosphate, sugar, phosphate, sugar, phosphate. You end up forming this long chain. So here's how it ends up looking. Here's the chain. So doesn't this kind of look like a protein? Doesn't it kind of look like there's the chain? In the protein, it was NCC, NCC, NCC. That formed kind of the chain. And then those R groups were hanging off. Well, in this one, the sugar connected to the phosphate, connected to the sugar, connected to the phosphate. So sugar, phosphate, sugar, phosphate. Hanging off of each sugar is a base. So this is kind of like written out a little more, but we can abbreviate it this way, where you just put a P and an S for sugar and phosphate. We can actually take and abbreviate it even more. So if you see it written like down at the bottom, that's just showing the bases. But remember, it doesn't mean that the bases are connected. It means that there's a sugar phosphate chain and the bases are hanging off. So can you see GTCA kind of going across the very top one? Then looking at the middle one, you can see it looks kind of like a shorter version. But then down at the bottom, they just don't put the sugar phosphate. But you got to just remember it's actually on there. So I'm talking about a chain, and those bases are like the ones that are hanging off. The other thing to notice, remember that proteins had a nitrogen end and a carbon end. So there was kind of like a beginning and an end. Notice it's the same thing here. There is, when they say this 5' or this little 5 comma end, that's the end where the phosphate is. Over on the sugar end, that's the end where the alcohol group is still on the sugar's carbon number 3. So that's where these numberings come from. There's always one end and another, and there you can't take and flip it. So the order of it, it goes 5', then phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then on the other end would be that 3' end. So there's kind of a beginning and an end to the strand, just like what you saw with proteins. But these are just kind of ways that they simplify, because you can agree the very bottom one is the fastest one to draw. If you just had to put the bases, you didn't have to write the sugar phosphate stuff, you just put the very bottom. So things that went on, and this, again, this is something that happened actually after the whole penicillin thing. This was in the 1930s, 1950s. So, Chargaff was the one, he was probably in 1940. So this guy came and he was like trying to study DNA. So they knew that nucleic acids, specifically DNA, had some kind of role. Like it was big. They found it in all of the nucleus of a cell. They saw uniformity. So they started like collecting DNA from frogs, collecting DNA from people, collecting DNA from other living cells, and looking at the bases. And what he found was, he found that A and T were always in equal amounts. So if you have 100 A's, there's 100 T's. If you had 400 G's, there's 400 C's. So he found that the amounts of A and T and the amounts of G and C were equal. But he didn't really know what that meant. He was just like, isn't that neat? Okay, and that was kind of the end of it. But in the 50s, Watson and Crick, who later ended up winning a Nobel Prize for this, they took this information along with information by other researchers. One of them was a woman, Rosalind Franklin. She was like one of those like super uptight chemists. She liked to do things like absolutely exact. So she figured out that she knew there was some kind of a helix involved in DNA, because she did what was called X-ray crystallography. So she like froze DNA molecules and then like took X-rays of them, which was like I said, she was like super detail oriented kind of person. They took all the data that people were publishing and they sat at a pub and drank beer all night and came up with, I think I've got it figured out. And they were right. They came up with the structure of DNA. They didn't actually do the research, but they were really good at taking like this information, this information, this information, this information, and going, well, this has to be that, and this fits here, like pooling it, putting it all together. So they came up with this idea that one, DNA is a double strand. That the opposite strands, wherever there's an A, it actually hydrogen bonds and fits with a T. They hydrogen bond, and that's what holds the two strands together like magnets. So you can peel it apart, but it comes right back together. They also found that the strands actually run in the opposite direction. So if one strand is from 5 to 3, the other strand's 3 to 5. So they go in this opposite direction. So they say they're anti-parallel. They published this and everybody was like, you're right. And they were like, yeah, it was a long night. But the strands run in opposite direction. So if I have 3 prime A, T, C, G, G, C, A, T, T, and then 5, what would be the complementary strand? What would start? What would this one be? This would be the 5. And so A would match with T. T would be A, G, C, 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 then G, T, A, A. And then over here would be the 3. So those strands run in opposite directions. Everywhere there's an A on the opposite strand's always a T. Everywhere there's a G, there's a C. And they actually hydrogen bond. And that's what holds those two strands together as a double strand. So things that we know now. So there's like what the strands look like. So things that we know now is DNA doesn't exist just as this loose strand. Because in fact, if you took all of the strands of DNA out of one nucleus and lined them up end to end to end to end, they would stretch out about 5 feet. Okay? So we're putting this into this microscopic little space called the nucleus. Can you see that that would cause problems? That things would get twisted? That I could imagine like, kind of like yarn. Like if your yarn doesn't stay wrapped, if it's just in a basket all loose, and you tried to pull one string out, it would be a holy mess. You would be all knotted together. I was trying to do this with my blasted gardening hose last night. So I was trying to stretch it out. I was trying to get it to roll up nice. It was like all bent over backward, like, ah! It's in the backyard, trying to stretch it out. So same thing. And that's just like a single garden hose. So you can imagine something super, super thin. It's like nanometers thick. But it actually would end up being 5 feet long. DNA has to have like more stability. A lot like proteins, remember like how the secondary structure gives you this stretchiness so that you can like stretch a protein and it bounces back? Well, there's the very top. So that blue shows you the double-stranded. Notice that it twists. So one, twisting is going to give you this stretchiness. Right? Just like those alpha helixes and beta pleated sheets. We've got some stretchiness to DNA too. But DNA actually wraps around proteins called histones. Histones, I think of them almost being like spools. So it's like, just like you would spool thread, you can also spool this. So the DNA gets spooled around these histones. And what it does is it causes the DNA to then start to coil back on itself. Coiling and coiling, that actually makes what they call supercoiled, because that is a coil of a coil with some coils. So that helps to like take this really thin, long strand, thicken it, and make it shorter and shorter and shorter and shorter, so that eventually you have a very short piece that's not going to get torn and it's not going to get knotted. It's much easier to manage. And when the cell goes through cell division, it actually wraps all the way up into a structure that they call chromosomes. So chromosomes are strands of DNA that are super wound. You only see chromosomes when the cell goes through cell division. So if you remember your A&P or biology, that's like during mitosis. That's when the chromosomes have to wrap up. And that's how they do it. And the only reason they do it is because otherwise it'd be a holy mess. They'd be all knotted up. Things would never divide equally. But this kind of like puts it almost like yarn. It's like now you got little skeins. Those are pretty easy to divide. If you didn't have them wrapped that way, it would really be very difficult to keep them from getting all knotted together. So they say it decreases the length by about 1 million and it increases the width by about 1 million. So now you got a nice short little compact structure. So I like this slide because this slide shows you kind of a compare and contrast. So here's the information of DNA. Here's the information for RNA. DNA is double-stranded. RNA, though, is only a single strand. DNA has deoxyribose. RNA has ribose. There's a difference. DNA has A, T, G, and C. RNA is A, C, G, and U. So it has a uracil instead of a thymine. DNA is much different than RNA by size as well. So DNA has 3 billion nucleotides, which is like almost amazing to think of. So like the base pairs, there's 3 billion of them in a cell. RNA is only made from DNA when it's needed because RNA is used to make a specific protein. So it's not present in the nucleus all the time. It just gets made so that the cell can make lots of one kind of protein. I think of DNA as being the complete set of genetic information. So like I said, it's everything that the cell needs to go from one single fertilized egg to being the size that you are today. Whereas RNA is only used to make one protein. So in the sort of grand flow of things, this is one of the big jobs that DNA has. It's not its only job, but one of the big jobs that DNA has is it's used to make a protein. Whether you're making collagen, or if you're making hemoglobin, or if you're making one of the many enzymes that you need to like digest starch or digest proteins or build muscle, whatever, that DNA gets converted to RNA, which then is converted into a specific protein. So I want to look at this and talk about this, because we're not going to talk about DNA at protein synthesis, but I want to think about this. So what happens if there's a change in your DNA? So any change in the sequence of DNA is called a mutation. So when they talk about mutation, that's what they mean. Mutation means there's a change in those 3 billion base pairs. So that's a lot. 3 billion base pairs. If you had to keep up with 3 billion little letters, okay, a change in any of those is considered a mutation. So possible effects. So if instead of an AT, it gets changed and now we have a GC in this one position. Possible effects, a lot of times, it'll have no effect. And if that's the case, they say it's a silent mutation. There's a lot of silent mutations. In fact, that's how your DNA can be taken, sequenced, and compared to other people's, and they're different. There's a lot of mutations that have happened over the many, many generations that humans have been around, so that there are differences between you and me. My DNA is going to be more similar to my parents, less similar to people that are not related to me. But even my siblings can have differences because we're all combinations of egg and sperm. So every egg and sperm is a little bit different. So every offspring's a little bit different as well. So we could say that families have certain DNA sequences and have certain mutations compared to others. But a lot of times those are actually just silent, which means that they don't have any effect. The problem can come into play, though, is if there's a change and it's in one of those sequence regions called a gene. Not this one, this one. If there's a mutation in a gene, this could change that whatever the gene is used for. So that might mean you don't make a protein. That might mean you don't make an enzyme that you need. That might mean that your cell doesn't do what it should. So if the mutation is in a gene, then the cell might, one, it might die, especially if it's a really important gene. If it's a really important protein. Two, the cell might develop a disease. So remember PKU? So remember I mentioned that again in the last chapter, the baby where they do a heel stick and test to see if you have this enzyme. So if you don't have that enzyme, that's because there was some kind of mutation that happened in that gene. Now you don't make that enzyme. Now we have to give you a special diet so that you have normal neurological development. Unfortunately, worst case scenario, well, I don't know, cells dying is pretty bad. Okay, so if the cell dies, if it's just that one cell, other cells might be able to pick up the slack. Disease, then you may end up having to have some kind of medical intervention for survival type of thing. But the other one is it may cause loss of cell growth control. So there are genes that specifically control how fast and how often your cells divide, right? So like your skin cells, if you go like this, like you do this all the time, you lose like 1 million skin cells a day. They just fall off. So you're like, that's how bloodhounds can track you, because you lose them, okay? But they're always growing from underneath. So your skin cells, they're replaced about every month. You have an entire new exterior, okay? Every month. It goes from the very bottom to the top layer and sloughs off in a month, okay? But then you have cells like nerve cells. They don't divide, okay? Cardiac muscle cells, they don't divide. But if you have a cell that lives in a certain place and has a certain job in your body, and you lose control over that cell growth, and that cell starts to grow faster than it should, what can that cause? Cancer, okay? So this can cause abnormal cell growth, might form a tumor, right? Because that's all a tumor is. A tumor is just a collection of cells that are growing faster than they should. But those cells may actually leave the original tumor and spread or metastasize and then form tumors in other organs. So these are kind of like effects that mutations can have. The cell might die because it could be a very important protein that's not getting made. You might end up with some kind of disease from it, or you may end up with abnormal cell growth. So then the next question, so what causes a mutation? So sources or causes of mutation. Most mutations are just random. They just happen. You have to take 3 billion base pairs and copy them for your cells to divide. So if you had to copy 3 billion letters, do you think you would ever make a mistake? Thank God it's not me, because if I was typing, it's like backspace, 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 backspace. Like I'm constantly, okay? It's kind of, I have autocorrect and it helps some, but I'm like, I know how to spell things, my fingers just don't work. That's the whole thing. So there's a lot of errors that just sometimes happen, especially when cells get ready to go through division. They have to copy the DNA before the cell can split into 2 cells. So you've got to actually make a whole copy, and you may have an error there. Most of those errors are actually fixed, but you can have what they call spontaneous mutations. If they don't get fixed, then they stay as part of that DNA for the rest of that cell's life. The other one, though, is you can have what are called mutagens. A mutagen is one that can cause mutation. So in this, a mutagen, you can have viruses that can act as mutagens. So viruses can cause changes in the DNA of a cell. Chemicals can cause changes in the DNA of the cell. The other one is energy. Those are sort of the ones that can cause mutation. Viruses can cause mutation. Chemicals. So notice there are some chemicals that they like have the skull and crossbones on. Like a lot of like the pesticides, insecticides, herbicides. All the sides, what do? Anything that's a side, it kills something, right? So a lot of those chemicals, not only do they kill the insect or the plant or the pest, they may possibly cause cell damage. And so that's where, like they say, to wear gloves, wear a respirator, limit your exposure. So you don't want to get it and come in contact. You don't want to ingest it, all because if it has the ability to destroy cells, then you don't want it to possibly get ingested and cause damage to your cells. Energy. Can you think of an example where energy increases your risk of cancer? Exposure to some kind of energy, sunlight. If you get repetitive sunburns as a child, that puts you at higher risk for developing skin cancer. When you get a sunburn, you have damaged those epithelial cells, and the damaged cells peel off. The cells underneath, they replace it, but those cells may have a mutation because of that exposure to that energy, and that can cause those cells to begin growing without normal control. So sunlight. Remember when we were talking about radiation in chapter 2, like Marie Curie, who had her little bottle of radium next to her bed? She died of leukemia. Okay, so radioactive energy is another one. Remember the people in Chernobyl, that they had a much higher incidence of cancer rates. It went up 60-fold in thyroid cancer, all because of that radiation exposure. So you want to try and limit like energy exposure, like insecticide, any of the side type of chemicals, and then viruses can cause that damage as well. Oh, well, we're done. So those were like the big things that I wanted to talk about. So on here, right? So we have basically gone all the way to the very bottom on this review sheet, all the way to 44. So we've talked about mutations, what causes mutations, what are possible effects of mutations. Okay? So we've covered everything. So the way that but Monday is your 5th exam. Okay? Monday, I have a note card. Do you have a note card? Do you need a note card? I have note cards. I brought my note cards. I just forgot to give them out. Okay? So don't forget to do your tape. That's not a pencil marks all over it. Okay. Herbicides. Like pesticides, insecticides, any of those, any kind of chemicals that have the skull and crossbones. So you can do your note card and it's over 9, 10, and what we covered tonight in 11. Okay? Just what we covered tonight in 11. Just the slides that we covered tonight in 11. So you don't have to worry about everything in 11, just the stuff we covered, which is really like the structure of DNA and RNA, and then mutations. Okay? That's Monday. All right.
And the test review going to be, is that updated or
I can do that. I can do that. Okay. Update, like pull out the questions that you don't have to worry so much about. But you just look in Moodle. I'll put it in exam 5. I'll list it as exam 5 updated review.
Okay.
Okay. Because nobody has to come to lab tonight. So that gives me some time so I can actually go do that over the next hour. So just look later tonight. Okay.